Ever pondered on the idea of editing the blueprint of life, our DNA, just like a text document? This might sound like a futuristic dream, but it's the reality of what we call DNA editing. This scientific marvel is all about making precise alterations to the DNA sequences in the cells of living organisms. Imagine a document on your computer. If you spot a typo, you can simply delete it, replace it, or even add something entirely new, all with just a few keystrokes. DNA editing works in much the same way, except the document here is the DNA molecule, and the typo could be a genetic mutation causing a disease. At the heart of this technology is a molecular tool called CRISPR-Cas9, which acts like a pair of molecular scissors. It has the ability to cut DNA at specific locations, allowing scientists to add, delete, or replace DNA bits. This remarkable technology has opened up a world of possibilities in various fields, from medicine and agriculture to environmental science and even biosecurity. Imagine the potential of this technology. You could potentially edit out genetic diseases from the DNA, making them a thing of the past. In agriculture, it could mean crops that are more resistant to diseases and pests, thereby increasing yield and reducing dependency on pesticides. The potential applications are immense and the implications profound. However, with great power comes great responsibility. As we hold the power to rewrite the very code of life, it's important to tread carefully. There are ethical considerations to be kept in mind, and potential risks to be mitigated. The science of DNA editing is still young, and there's a lot we don't know about the long-term effects of making changes to the genome. Indeed, while it sounds like a plot from a science fiction movie, DNA editing is a real and powerful tool in modern biology. It's a testament to the astounding progress we've made in understanding the secrets of life and our ability to manipulate them for the betterment of our world. One of the most revolutionary tools in DNA editing is known as CRISPR-Cas9. This powerful genetic tool is a game changer, a revolutionary leap in our ability to modify the very building blocks of life. The story of CRISPR-Cas9 begins with a simple observation in nature. In the early 90s, scientists studying bacteria stumbled upon a peculiar genetic sequence, a series of repeating DNA segments separated by spacers. These spacers, it turned out, were snippets of viral DNA, a kind of genetic memory of past infections. This discovery led to the identification of the CRISPR system, bacteria's defense mechanism against viruses. Fast forward to 2012, when two researchers, Jennifer Doudna and Emmanuel Charpentier, discovered that this system could be harnessed as a tool for DNA editing, and the CRISPR-Cas9 technology was born. But how does it work? Imagine a pair of molecular scissors capable of cutting DNA strands with astonishing precision. That's essentially what CRISPR-Cas9 is. The CRISPR part of the system is a guide made of RNA, the cousin of DNA, that directs the Cas9 enzyme, our molecular scissors, to the exact location on the DNA strand that needs editing. Once there, Cas9 makes a precise cut, and the cell's own repair machinery kicks in to fix the break, allowing scientists to add, delete, or alter specific genetic information. This level of control over genetic material is unprecedented and opens up a world of possibilities. It allows us to fix genetic errors that cause disease, engineer crops to withstand harsh climates, and even bring back extinct species. However, with great power comes great responsibility. The potential to rewrite life's code carries profound implications. We're not just talking about scientific challenges here, but ethical ones as well. What are the boundaries? Who gets to decide what modifications are acceptable? CRISPR-Cas9 has indeed opened new horizons in genetic research, but it also brings with it a set of ethical considerations. As we continue to explore the potential of this revolutionary tool, these are questions we must grapple with, ensuring that our reach does not exceed our grasp. As with any powerful technology, DNA editing carries with it certain ethical considerations. DNA editing, as awe-inspiring as it may be, is not without its share of potential pitfalls and contentious issues. These ethical concerns are as intricate and complex as the DNA helix itself. Firstly, let's consider the potential for misuse. While DNA editing holds the promise of curing genetic diseases, it also has the potential to be used unethically. Imagine if DNA editing tools fell into the wrong hands. The creation of designer babies, where parents could potentially choose their child's physical traits or abilities, could become a reality. This not only raises questions about the sanctity of human life, but also about the potential for a new form of eugenics. Next, we have the risk of creating inequalities. If DNA editing technologies are only accessible to the wealthy, we could see a widening gap between the genetically enhanced and the rest of society. This could lead to a new kind of discrimination, a genetic class divide, if you will. Then there's the question of consent. Who gets to decide whether a person's genes should be edited? If we're talking about editing the genes of an unborn child, for instance, they obviously can't give their consent. And what about the potential long-term effects of gene editing? How do we navigate the murky waters of consent when the full implications of DNA editing are still not fully understood? Lastly, there's the issue of playing God. Some argue that meddling with our genetic makeup is overstepping the bounds of what is morally and ethically acceptable. They contend that we are tampering with the very essence of what makes us human. Balancing the potential benefits of DNA editing with these ethical considerations is a complex challenge that society must address. As we stand on the precipice of a new genetic era, we must tread carefully, ensuring that our desire to cure does not overshadow our responsibility to care. Despite these concerns, DNA editing holds immense promise in the field of medicine. One of the most compelling applications is in the treatment of genetic diseases. For centuries, these conditions have confounded doctors and caused untold suffering for patients. But with DNA editing, we might finally have a potent weapon to combat them. Imagine a world where a child diagnosed with cystic fibrosis could receive a single curative treatment that edits the faulty gene responsible. Or consider the potential for people with sickle cell anemia, a painful and debilitating condition caused by a single genetic mutation. With DNA editing, we could correct the mutation, offering these patients a new lease on life. But it's not just genetic diseases that could benefit. DNA editing also offers great promise in the fight against cancer. Traditional treatments like chemotherapy and radiation often have severe side effects because they can't distinguish between healthy and cancerous cells. DNA editing could change this. Scientists are developing ways to use CRISPR, a revolutionary DNA editing tool, to precisely target and kill cancer cells while leaving healthy cells untouched. Beyond cancer and genetic diseases, DNA editing could also revolutionize the way we develop new drugs. By making precise changes to genes, researchers can gain a better understanding of how diseases develop and how they can be stopped. This could lead to the development of more effective treatments with fewer side effects. Of course, it's important to note that we're still in the early days of DNA editing in medicine. There's much we don't know, and there are significant ethical and safety considerations to address. But the potential is there. If we can navigate these challenges, DNA editing could transform medicine, offering hope to millions of patients around the world. While still in its early stages, the potential of DNA editing in medicine is truly exciting. So, where are we headed in the realm of DNA editing? As we move forward, the future of DNA editing technologies seems to be brighter and more promising than ever. The advancements in this field are ushering in a new era of precision and efficiency, and the possibilities are simply astounding. Consider the development of more precise and efficient editing tools. The next generation of DNA editing technology is set to be more accurate, faster, and safer. Researchers are working tirelessly to improve the precision of these tools, reducing off-target effects and increasing the specificity of gene edits. This means that in the future, we may be able to edit genes with pinpoint accuracy, minimizing risks and maximizing benefits. Then there's the potential for gene therapy. DNA editing technologies could revolutionize the world of medicine by making it possible to treat or even cure genetic diseases at their source. Imagine a world where conditions like cystic fibrosis, muscular dystrophy, or sickle cell disease could be treated with a single procedure, altering the faulty genes and giving patients a new lease on life. This isn't just a dream, but a tangible goal within our grasp. But it's not all smooth sailing. The road to the future is fraught with challenges. Ethical considerations, regulatory hurdles, and technical limitations all pose significant obstacles. It's a delicate balance, ensuring that the benefits of these technologies outweigh the potential risks. We must navigate these challenges with care, ensuring that the science progresses in a responsible and ethical manner. Yet, despite these hurdles, the future of DNA editing technologies remains incredibly exciting. The dawn of a new age in genetic medicine is upon us, and the possibilities are as vast as the human genome itself. As we continue to push the boundaries of what's possible, we are unlocking the secrets of life at its most fundamental level. As we continue to explore and refine these technologies, the future of DNA editing remains a thrilling prospect.